Okay, cool.
Well, hello everyone. Welcome to another session of our Evolution Seminar Series. We're very excited today to have Lauren Cirino, a postdoc from UW-Milwaukee in Rafael Rodriguez's lab, who's going to talk today about sexual selection. So please join me in a warm welcome for Lauren Cirino.
Let me turn this on. Just make sure y'all can hear me. So thank y'all for coming to my seminar today. I know the rain kind of put a little bit of a damper on actually coming physically to this talk, but I'm super excited to be here. I haven't visited UW-Madison yet, so this is really exciting for me, just to zip over from Milwaukee and be with you all today. So thank you so much for coming. And kind of on the thank you note, I just wanted to first thank both of my previous lab at University of Florida and my current lab members for just really helping me kind of flesh out some of the experiment ideas that I've had in the past few years through grad school and then into my postdoc at UW-Milwaukee. They've been really helpful in just making everything happen. And I would be absolutely nowhere if I did not have my amazing undergraduate student researchers that have helped me as well. They have pushed me to be a better scientist and a better educator, and they have really just enriched my time in both grad school and as a postdoc in Milwaukee. And so I've mentored many students. The experiments I'm going to talk to you today is just some of the experiments that we have done together. So this laundry list is for all of the experiments that I've done, not just the ones that I'm going to talk to you today about. Good? Cool. Okay, so sexual selection arises from competition between mates. And this can manifest in two different ways. It can manifest in indirect competition, where males have some kind of flashy or desirable trait to woo a female to mate with him. Or it can manifest through direct competition, where males engage with other males in head-to-head competition for access to territory and eventually mate with females. And so these types of competitions can lead to the extreme sexual dimorphism that we see between males and females in the animal kingdom. And also, it can lead to the elaboration of these traits. So in this peacock picture here, we can see the brightly colored male with his big peacock feathers. They also do like this little dance that's great to woo a female. And the pronged horns of the deer have also increased in elaboration over time. And these two types of competition live in this pre-copulatory space. So basically behaviors or traits that occur before copulation. But once a male achieves copulatory success, that isn't actually the end. It doesn't guarantee a male fertilization success. There is also such things as post-copulatory competition. So that's once the sperm is in the reproductive tract, what happens then? Well, cryptic female choice within the reproductive tract, so where females are choosing between sperm of different males and then accepts a subset of those sperm to fertilize her eggs. That's cryptic female choice. And there's also sperm competition, where sperm from some males, from one male is competing with sperm from other males. And so this can also happen post-copulatory. So once they kind of go through all of this, these different bits of competition, they may hopefully achieve fertilization success. And so all of these traits and behaviors kind of all fall into different mating systems. And so the mode of competition can lead to a mating system, and then a mating system can reinforce competition. And so that's how we think about it and what's out there in the literature right now. This is how it's kind of designed, these mating systems. And My research kind of falls into the pre-copulatory context that we were just talking about, so the female mate choice and male-male competition. And so I'm really interested in the reproductive consequences of pre-copulatory competition in species with different mating systems. And the two species that I will talk about today that have two different mating systems are the tree hoppers. So sorry about that one's so small. That's a tree hopper right there. And the leaf-footed bug, that's a silhouette of a leaf-footed bug there. You'll see more fancy photos in a little bit. And the tree hoppers are monandrous. So that means females mate once and then they're done. And males can mate multiply. And the leaf-footed bugs, these guys over here, they are polygamous. So both females and males mate multiply. And so we're gonna kind of peruse through pre-copulatory competition with both tree hoppers and leaf-footed bugs today. And we're gonna start with tree hoppers. Maybe. It's okay. Got it. Okay. So widespread in the animal kingdom are Darwinian sex roles, and Darwinian sex roles arise through resources being partitioned slightly differently. So in males, resources reproductively are partitioned towards a lot, like many tiny sperm. Whereas females will partition those same resources to a lot fewer but larger eggs. So it's the same amount of resources are being devoted to gametes, but just in different ways. And so under Darwinian sex roles, males should be mating very quickly with females and mating multiply to increase their overall fitness, because they have lots of little sperm that can fertilize lots of eggs. So they should be mating with females, mating with females, mating with females, mating with females. However, we see across the animal kingdom that males are prolonging copulation. So they are engaging in a copulation and it's taking hours before they go off to search for another mate. And I'm like, why? Why would you do that? If this is true, why would you prolong a copulation? You want to have enough time so you'll fertilize her and then move on and search for another female and gain as many matings as possible. So there are some reasons or some hypotheses for reasons why prolonged copulations happen. And one is just the very fact that sperm may take a long time to transfer. So that sperm may just take a really long time to transfer, or there may be an interval that males have to wait upon copulation before they actually are allowed or can actually pass their sperm along. Not mutually exclusive are these three other hypotheses. So sperm does need to go to eggs, but males provide an entire ejaculate, which has sperm and other accessory substances to eggs. So perhaps both sperm and the accessory substances take time to move through the reproductive tract. And these three hypotheses say slightly different reasons for the accessory substances. So the receptivity reduction hypothesis says that accessory substances are transferred so that the female will only mate once, or reduce receptivity, and she won't have to mate with, or she won't mate with another male. But alternatively, the accessory substances could be used as a nutritional gift. And so passing along accessory substances that allow females to have extra resources to perhaps live a longer life. And if she lives a longer life, she's able to lay more eggs and produce more offspring that way. And finally, the life history manipulation. So upon copulation and passing accessory substances, perhaps this upregulates reproduction. And so she lays lots and lots of eggs really quickly, but because she's devoted so many resources to reproduction, that's going to curtail survivorship. So this is a life history manipulation, so survivorship is traded off with reproduction. Okay, so this makes sense in a polygamous or a polyandrous, so females mating multiply, it makes sense in this situation, this mating system. Females are going to mate with lots of other males. So having these different accessory substances and prolonging populations with females makes sense. But what doesn't make sense, and was curious, is that monandrous species also prolong copulations, but females only mate once. So I wanted to know why. Why is this happening? Why? So to do this, we used a species of tree hopper, Echinopha binatata, and they are, as I said, monandrous. They only mate once. Females only mate once. Males can mate multiply. And they engage in prolonged copulations. They can, sorry, they can mate for up to 3 hours, which is, if you are a behavioral ecologist studying mating behavior, watching them for 3 hours is boring. But we do it for science. And finally, and something really cool about this species is that they engage in substrate-borne vibrational communication. So males will vibrate a tree branch at a given frequency. And if that frequency is really attractive and sexy, a female will respond with her characteristic response. Now the males have a sexier vibration than the females. The females kind of, and it's something like this, maybe. Oh, nope, we're gonna try. Nope, we're gonna try. We're gonna make it happen. It was just doing it. Oh, no. It's so good. I don't wanna. No, okay.
Maybe the USB.
Well, that's okay. I mean, I can also do it aloud for your ears. My voice. So the males have a, and the females on each time point will go, and so this duet with a, that's a duet because the males and the females are both signaling to each other. They do that until they finally form pairs, and they usually sometimes copulate. That was always a question mark whether they were doing that. Okay, so we placed the tree hoppers into 5 different mating duration treatments, anywhere from 5 minutes to 60 minutes. And then we had an uninterrupted group that we just allowed to mate as long as they wanted to. And that acted as our control treatment. And the average copulation duration for that group was 104 minutes. So it did last longer than the 60-minute treatment. And then we measured female receptivity. So we played them a playback. So we played them a male signal, and we listened. They don't feel it, but we hear it in our recordings. We were waiting to hear a female response. And if they responded, great, they were still receptive. If they didn't, they were not receptive. And then we also allowed them to live out their entire lives on a tree. So we tracked their survivorship. And then we looked to see whether or not they were laying eggs. If they laid an egg or not, we quantified that. And then the species is univoltine, so they only produce 1 generation a year. And then their eggs overwinter. And then in the spring, we counted the number of offspring. And so I just wanted to give a big shout out to Sage and Ariel. They were my 2 undergraduate research assistants for this project and helped me kind of quantify all of this data, because we were tracking survivorship every other day to make sure that, to see whether or not females were still alive. And then we were also counting eggs. So, sorry. So before we get into the data here, I just want to orient you to this graph. So on the x-axis, we have our mating duration treatment. And on the y-axis, this is whether or not the reproductive event happens. And on this graph, you will see 3 reproductive events. So receptivity, did that happen? Yes or no? So did a female respond? Eggs, did they produce at least 1 egg mass? Yes, no. And finally, offspring. Did at least one juvenile hatch from the egg mass? Yes, no. So all of these are yes, no data on the y-axis. So what we found was shorter mating duration treatments. Females were receptive to those male signals that we were sending them. So they were more likely to be receptive and compared to the uninterrupted and longer treatments. In terms of eggs, the shorter mating duration treatments were less likely to produce egg masses, whereas the longer mating duration treatments were more likely to produce egg masses. And although this was not statistically significant, we all have eyes that we can see. 10-minute females did not produce a single offspring. So that's 0 compared to those that were in the later mating or the longer mating duration treatments of 60 minutes and uninterrupted. So they were actually producing offspring. And because this wasn't statistically significant, and I'm like, no, but you can, there's a pattern and you can see it, right? I then looked at and quantified total offspring produced. So the actual numbers, how many of them did they produce? And we found that in the 60-minute and uninterrupted treatments, they produced similar amounts of offspring compared to the almost none in the other 3 treatments. So the 30-minute and 45-minute treatments, only 3 females produced offspring. So to tackle the other 2 hypotheses that I mentioned at the top of this part of the talk, the life history manipulation hypothesis and the nutritional gifts hypothesis, we tracked survivorship. Before we looked at how mating duration affected survivorship, we just wanted to make sure that the longer that females lived, the more offspring that they produced. So we assumed that, but we weren't 100% sure, so we just checked that. And that's what that inset graph is showing. So yes, the longer females live, the more offspring they produce. And then we looked at mating duration. Does that affect survivorship? And we found that it does not. Okay, so what does this all mean? So coming back to these hypotheses, we found evidence for the sperm transfer hypothesis and the receptivity reduction hypothesis, and we rejected the other 2. So the sperm transfer hypothesis, so it takes tree hoppers about 60 minutes to actually transfer enough sperm for many of them to start producing offspring. And as far as receptivity reduction goes, well, it takes over 60 minutes for males to pass those accessory substances to reduce receptivity, preventing them from mating again. So essentially, this process, this prolonged mating duration, makes this species a monandrous mating system. I just wanted to give a shout out to Ariel. He gave this presentation, or he presented these results at a couple of symposia in the spring and did an awesome job. Okay, so we come back to our this is our overall slide. This is going to be a flow chart soon. So we have our tree hoppers, and I did not present any data on the direct competition, so male-male competition, but I will tell you they don't actually like face off head to head. They're not engaging in fights, but they do some jamming. So those signals that they produce for females to respond, sometimes other males will overhear those signals and they'll jam them. So females are like, ugh, that signal is now ugly, not attractive. So there is some competition going on, direct competition going on. But what actually is driving the traits of this species is female mate choice. It's this indirect competition. And there is, and the attractive traits are the vibrational communication, that whole vibrational communication system. And so this, in this species, female mate choice is really, really, really strong. In fact, even if a female is responding to the male, it can take over an hour for them to actually mate, which again, is just thrilling for all of us that were watching behavior trials. So prolonged copulations then are likely to evolve for the reasons that we just talked about, right? Sperm needs to be transferred, and then accessory substances are going to transfer because the males have just put in tons and tons of work to obtain a female mate. They don't want to have to compete anymore. My God, it takes over an hour to get a female. So post-copulatory, they are unlikely to have to deal with cryptic female choice because female mate choice, pre-copulatory female mate choice is so strong, and they're unlikely to deal with sperm competition because females are really not receptive to any other males in the population. Okay, so that's tree hoppers. So we're gonna move on to leaf-footed bugs, and remember, they are polygamous, so females and males mate multiply. So environments are extremely dynamic. They vary across both space and time. And we know that a poor early life environment for females can have strong negative effects on reproduction in their adult life. And some studies have looked at how an improved adult diet can influence reproductive traits. And there are 3 that I found. And those that have done this have found some reproductive rescue in both ovaries and egg numbers. But even though these studies did a really good job in helping us understand how reproduction can be recovered at adulthood, these studies didn't necessarily use the diets that these females would eat in the wild. And so we may be missing some information in how reproductive rescue can happen. So I wanted to actually look at this reproductive rescue in the leaf-footed bug using their actual diet. And I tested 2 hypotheses. So I tested the silver spoon hypothesis that states females raised under poor conditions are reproductively limited compared to females raised under good conditions, regardless of their adult environment. So what's happened? There we go. So what this means is if a female is fed an improved diet, it does do better than those that are fed a restricted diet, but it cannot outperform those fed a good diet or those that were fed from a silver spoon their entire lives. Hence the silver spoon hypothesis. Alternatively, females can fully reproductively compensate when the adult environment improves. So this means that the improved diet females are going to perform just as well as those fed a good diet. And again, so with a natural diet, that natural diet usually changes across time over the seasons. But not only does diet change, other ecological factors like temperature and photoperiod also change. So I also included those into the analyses of this study. And my overall question incorporates those as well. So I'm looking at how do seasonal diets affect female reproductive success across a long breeding season where things like diet, temperature, photoperiod are all changing at the same time. And to do this, I used the leaf-footed cactus bug, Narnia ephemerata, and it lives on prickly pear cactus and feeds on prickly pear cactus. And juveniles and adults live on the same food, so they all live on the cactus together. And the difference here is the juveniles, they don't have fully formed wings yet. So their mobility is restricted to just walking around to find food. Whereas adults have fully formed wings. So if the cactus patch that they were on is not so great, they can fly off to find better cacti. And we know that this cactus changes across the seasons. So early on, there are fruited cacti and they mature into this like fully red, it's really pretty, cactus. But in the autumn and winter months, the other herbivores out there, as you can see up here, are also feeding on this cactus fruit. And and the rest of the cactus fruit that's out there eventually falls down to the ground and also can decompose. So eventually this cactus fruit disappears. And this species is, this species has several generations per year in Florida. And so juveniles are subjected to fruit and no fruit cactus pads at different times of the year. And we know from previous work on this species that a cactus pad with fruit is really optimal, and that a cactus pad alone is really suboptimal. And previous studies have found that those that are fed the cactus pad with fruit grow larger bodies, males have larger testes, females produce more eggs. And in a previous study that I ran, we know even a change in cactus fruit quality, so from unripe to ripe fruit, can have rescuing capabilities for reproduction. So females are able to partially rescue the number of eggs that they can produce. So that really makes the species is a really ideal one to test this question. And so what we did was we placed juveniles onto 2 different diets. We placed them on a cactus pad with fruit and a cactus pad alone, so no fruit. And we allowed them to mature into adults and continue to feed on this diet. We then took a subset of those that were fed the cactus pad alone, the cactus pad without any fruit diet, and we placed them on cactus pads with fruit as adults. And so from here on out, we'll call these the fruited diet, the restricted diet, and the improved diet. And as I said, we did this over a long breeding season. So we tracked females for 9 months, and temperature and photo period did change, even though we had all of our bugs in a greenhouse with the lights on a 14-10 light dark schedule. We still had the sunlight changing and the temperatures changing outside. So things changed. And so again, we factored that into our analyses. We also looked at them at 2 different ages. So these bugs become sexually mature at 2 and 3 weeks of adult life. So we assessed them at those 2 time points. And what we were looking at was mating behavior. So they have a ritualized mating behavior where a male will mount a female on her back, so dorso-ventrally, and they will put their genitalia at the very tips here. And then if a female accepts that male, the male will turn 180 degrees, kind of like this, and they will continue to mate. If not, she can close her genital plates, she can kick that male off, she can run away. I've seen it all. So much rejection. And so that made it really easy for us to track the behavior and track female receptivity, which was what we're looking for here. And this, the bugs were put into a deli cup environment. So we had just a big old deli cup with a cactus pad planted in it. And the males were placed in the deli cup with the females' adult diet. And I'm going to show you several graphs. They're all going to look the same. So we've got diets in this left graph, age in the middle graph, and time in breeding season on the right graph. And the y-axis here is likelihood for a female to accept a mate or female receptivity. So it's just a yes, no, again, analysis. And so what we found, surprisingly, was there was no difference in female receptivity when her condition differed. So whether she was feeding on the restricted diet or the good diet, that didn't change her receptivity. However, when she was older, when females were older, they were more likely to accept mates. And also time and breeding season did not affect mating behavior. So really the only thing that was affecting mating behavior here was age. And then I just wanted to make sure that my previous results were replicated. here. So I also noted whether or not they had eggs in their reproductive tract. So after the behavior trials happened, I dissected them and looked to see if they had eggs in their bodies. Again, a yes/no analysis there on the y-axis. And what I found was, again, even though not statistically significant, you can see the improved diet females. There were at least a few that started producing and yolking up eggs. Perhaps they needed more time to actually start yolking up eggs in that group. Age also affected whether or not they had eggs in their reproductive tract. So older females were more likely to have those eggs. And then time in breeding season was a huge factor. So earlier in the breeding season, females were more likely to have eggs. Later in the breeding season, less likely. So kind of all of these things are working towards egg production here. Okay, so just to remind you about our hypotheses, silver spoon hypothesis says that the improved diet females will partially be rescued in their reproductive traits, but not able to outperform the good diet females. The alternative hypothesis just states that females will do just as well as the good diet females. And so my egg data states-ish that they are able, again, we have this improved diet female group here. They're able to, some of them were able to start producing eggs, and likely more of them would as time went on, and they were able to acquire more resources to put towards reproduction. So silver spoon hypothesis is supported here. But really what I was focusing on is mating behavior. And it was just weird, right? Like they didn't change their mating behavior. Even the restricted diet females, the ones that were fed the worst possible diet, they were still mating. And honestly, good for them, right? So what is going on here? Possibly, these ecological factors aren't really affecting mating behavior because females have a sperm storage organ in their bodies. So they can store sperm so that when they have one egg, they can fertilize it right away, and they are not gonna waste any time trying to find a male to mate with. So this might be the reason why mating behavior doesn't change. Okay, so this data that I showed you right now was just like a little nugget of data. This project was one of 3 projects that lasted 9 months, and like all of these other incredible stats. And I would never have been able to do that without these undergraduate students. They were absolutely amazing and really a joy to mentor. And so I just wanted to give them a huge shout out because they were so helpful in this project. Okay, so back to our little flow chart here. We've got our leaf-footed bug and indirect competition. So remember, that's female mate choice. So female receptivity. We see that there isn't really a strong female mate choice component going on based on my research and actually based on other research previous to mine as well. Female mate choice is just pretty weak in this species. But what about direct competition? So males use their sexually selected weapons to engage in male-male competition, and they do this to win territories and access females. But not all males can grow the biggest, most impressive weapons. You can see there's a lot of variation in weapon size here between just these 3 species. And if weapons are small, males are more likely to be subordinate to males with larger weapons. And so they have to kind of figure out another way to perhaps achieve mating success. But even if males that grow the biggest, largest weapons, even those males may experience weapon damage. And while weapon damage is pretty common across animal taxa, what is less common is for people to research what the heck happens with fertilization success. So I did it. And I used Narnia ephemerata again, so the leaf-footed bug. And the reason I chose this species is because they have sexually dimorphic hind legs. Those hind legs are wide, and they have little spines on them. And they use them to engage in male-male contests, where both of those weapons, those hind leg weapons together, make up a complete weapon that they use to kick and grapple and squeeze other males so that they can be dominant on the cactus pad territory. And they also perform this really cool anti-predatory behavior called autotomy, or self-induced limb loss. And this occurs, it's pretty common in the species. About 12% of the time we see individuals with an autotomized limb. And the reason for this is anti-predatory, as I said. So if a predator comes and nabs them, they nab one of those big legs, right? What they can do is they can break off their leg at the trochanter femur joint, and they can escape with 5 legs. They can also get out of a fouled molt, and this is pretty common. So the juveniles will like get out of their little molts, yeah, doing it, and then it's, it's stuck. And it's stuck on their leg. And so instead of like dragging that leg around, they will just break it off at the trochanter femur joint, and 5 legs, no problem, can live my life. Or can they? Because those males with a missing hind leg, an incomplete weapon, are 5 times less likely to become dominant on a territory, which is nuts. So they're way more likely to be subordinate. And oh man, what happens to mating success? Well, females, like I said, weak female mate choice, females don't appear to care. Complete weapon, not a complete weapon, no problem. I'll mate with you, which is great. So males may be able to still mate with those types of males. And we know from previous work that if males experience weapon damage during the juvenile stage, they will grow larger testes and produce more sperm. And yes, those are actually testes of the leaf-footed bug. Those aren't just like fancy little hearts. So it was really nice to dissect these bugs, and it was so easy to pluck out. Oh, yep, there's a testes, it's red. And so this here supports the reproductive trade-off hypothesis. But I wanted to know, okay, so yeah, you're growing bigger testes, you're producing more sperm, but can you actually produce more offspring than intact males? So my undergrads, Brianna and Haley and I, we worked together and we predicted that weapon damaged males would produce more offspring. So what we did was we induced weapon loss at the juvenile stage. And so we placed some males in the weapon damage treatment and some males in the intact control treatment. And then we placed males with females in their little deli cup environment for 24 hours to mate or not mate. And then we took them out, placed them in another cup for 24 hours with them, mate or not mate. We did that for 4 days straight with 4 different females. And we placed them in a non-competitive environment, so we knew who the offspring were coming from, right? And competition brings in a whole other ball of wax. So non-competitive environment, so just 1 male and 1 female. And then we allowed those females 2 weeks to lay eggs. And then we allowed those eggs to hatch for another 2 weeks. Both of those timelines is characteristic for this species. And then we quantified eggs, hatching success, and offspring numbers. So in this first graph, we have the type of male that the female is mated with, so weapon damaged or intact control. And then on the y-axis, we have probability of eggs being laid. Again, a yes-no analysis. And we found that there was no difference between treatments. And the reason for this is probably because females in the species, they can lay unfertilized eggs. So what we then did was we said, okay, of those females that laid eggs, did weapon damage or intact control, did they increase the probability for hatching success? So that's what is on the y-axis. So again, this is a yes-no analysis. And we found that weapon damage females that mated with weapon damage males were more likely to have hatching success than in the intact control females. And finally, the money question, the question we were looking to answer, is the number of offspring. So we have number of offspring produced on the y-axis, and here is the female partner body size. We ran a PC analysis with a bunch of linear body size measurements of females, and PC1 came out as explaining the most variation in the body size. So that's what these numbers are on the x-axis. If you don't know what the heck a PCA is, don't worry about it. Small females, large females, done. In the legend, you'll see when the data pops up, the closed circles and the solid line represent the females that mated with weapon damage males. And the open triangle and the hash line represent the females mated with the intact control males. So what we found was that weapon damage males produced more offspring with large females than intact males. I'm just gonna stay on this graph for a minute. So over here, that solid line is the females that mated with weapon damage males. They're producing more offspring. This hashed line is the intact control male. And then over here, that pattern disappears with smaller females. And the reason that this might be is because large females in arthropods and insects are arthropods. They are more fecund the larger they are. So males are also really into that. And so this pattern disappears likely because smaller females can't produce the same amount of eggs. Okay, so if we look at this pie chart as one male is going to consume resources, and the pieces of the pie chart represent important life history traits. And so resources go to these important life history traits, including reproduction. And then once in reproduction, they have to partition them to pre-copulatory traits such as weapons and post-copulatory traits such as sperm. And if males incur weapon damage during the juvenile stage, this frees up resources that they can put to post-copulatory traits such as sperm, which can then produce lots of offspring, supporting the reproductive trade-off hypothesis. And so I just wanted to give a big shout out to Haley. Not only did she present this data at a bunch of conferences and symposia, she also helped write the paper, which is out in behavioral ecology and sociobiology, if you're interested. Okay, coming back to our flow chart. So direct competition appears to be really, really big in this species. There are traits that have evolved to help males win male-male contests and acquire territory and mates. But if weapons are damaged, they can access this other thing that has evolved, this reproductive trade-off that has evolved. And if they are maybe sly about it, they can mate with one female or a couple of females that those females don't care about whether they're weapon damaged or not. They can still achieve some reproductive success, or at least copulatory success. And that's kind of where I want to go in the future. So if these weapon damaged males have more sperm and they give the female more sperm, perhaps they're better at engaging in post-copulatory competition, like sperm competition. Maybe they can displace other males' sperm, or maybe they can just outcompete them. They may just be better sperm. And maybe this better sperm could be attractive to females post-copulatory, and she's choosing that sperm to fertilize her eggs. So that's kind of the next step, or where I'd like to go next with the leaf-footed bugs. Okay, so just as a quick reminder, tree hoppers are monandrous, so females mate once, males mate multiply, and leaf-footed bugs are polygamous. Females and males mate multiply. And as I mentioned at the top of this talk, the mode of competition, or kind of what's in the literature, is, the mode of competition is influencing the mating system's influencing the mode of competition, so on and so forth. And a lot of what's out there is saying that ecological variables are really controlling what the mating system is. So like habitat limitation is defining a mating system. But I don't think so. I'm gonna share with you what I think is going on. So in tree hoppers, we have strong female mate choice and really low likelihood of copulatory success. Remember, males have to put in a lot of work to get females to choose them and mate with them. That leads to indirect competition evolving and tactics to prevent post-copulatory competition, which means that this system is monandrous. So all of this competition and female mate choice is influencing the mating system, and then that mating system reinforces the top. In leaf-footed bugs, we have weak female mate choice and high likelihood of copulatory success, which means direct competition evolves in both pre-copulatory contexts and post-copulatory contexts. And the mating system is therefore polygamous. And then that's reinforced. So I pose that females set the rules and everything else follows from there. And that's how mating systems are determined. Here we are. This is what I suggest. And that's all. If you guys have any questions, thank you so much for coming. I'm happy to answer them. Yeah.
Do you know anything about in the footed male or females? Do they have sperm storage, like organs specifically for sperm storage?
In males?
Sorry, in females.
In females, yes. Yeah. In fact, both of these species do have some to some extent sperm storage.
Do you know how long they can use fertilized sperm from those organs?
Yeah, so not in these species specifically. And of course, my memory is failing me right now on what particular species, but there is a study that looks at sperm storage overwintering with sperm in their bodies, and they produce less offspring than they would if they had used it right away. But they still have some sperm. So they think that they are losing some of that sperm, or that sperm may not be functioning as much anymore, perhaps, but they're able to use at least some of it to get reproduction going right away, especially when you're coming out of the winter time and it's hopefully other males are around and they survive, but if they didn't, then boom, you have sperm that you can use to fertilize your eggs right away. So yes, there is some evidence in an insect that I can't remember. I'm very sorry.
Yeah, so I'm assuming if you haven't looked at this yet, then you don't know that much about this question, but
yeah,
it would be interesting to see if there's any preferences for mating order in terms of sperm usage. That's the thing in Buffalo.
yeah,
and if there's difference, just talk about different organs for sperm storage, and they store these. People don't know exactly what is stored where, but
yeah.
it would be curious if there's similarities between those or not.
Yeah, I in all of my research on this, or all of my paper reading on this, we think it's a first and last out sperm storage situation, but we don't know in this particular species. But that is a really cool and interesting avenue of future research that I would like to do. So I'm right there with you. Yeah.
I was wondering, do you know more kind of thinking in a comparative sense about these two different mating systems and their evolution? Is menandry ancestral in this kind of among these other closely related species and versus polygamy? So how do we think about these species and the evolution of their mating systems in a phylogenetic context?
I can tell you that in tree hoppers, in closely related tree hoppers just south of here, they are polygamous. So I'm not sure in the phylogenetic sense. That would be really cool to look at too. This idea that I posed today is in its baby stage of thinking.
I think it might really be influenced by what's happening in other species.
Yeah, totally. And also, we haven't really thought about it a lot in insects either. A lot in invertebrates and everything, looking at mating systems and how different, ecological factors, for example, affect mating systems, but not as many studies in insects specifically. So that would be really cool.
Like Parker and Thornhill, like mating systems and insects have been kind of the foundation of.
Oh, yeah. But like a consensus of like what is causing these mating systems overall is not really as much or as much in insects as, I mean, yes, I have looked at, I have a whole insect mating systems book too that I've been looking at too. But this idea has been like just kind of noodling around. So baby stages.
Do you know anything about the presence or importance of like pheromones or particular hydrocarbons in these species? Are those relevant?
Yeah, in the leaf-footed bugs, I did a study when I was a master's student on olfactory cues, and the males that were raised on the ripe fruited cactus versus males that were raised on unripe fruit cactus are producing different types of pheromones. So females are honing in on olfactory cues, and I assume cuticular hydrocarbons are also important, but we just don't know about those in the species. Oh, I'm sorry.
As a microbiologist,
Yeah, so I know that there is a leaf-footed bug where they studied the microbiome and they picked up bacteria horizontally. So they picked it up from their diet. So that may influence some of these traits as well. They are living on the same cactus pads, and they do feed on the cactus pads. Like the cactus pad is a really, really bad diet, and it has like mostly composed of water, has a little bit of carbohydrates, like just not a ton of nutrition the way the fruit has or what the fruit has in it. So I don't know if different bacteria live on those two different parts of the cactus even. I guess I just assume like it's all there, but that's a really bad assumption. So I think that they're definitely picking up something from their diet. I just don't know if it differs across time. So many good avenues of future research.
I may have missed this, but in your later experiment, the one where you were showing where one female or one male to one female, then to another female, were those females I guess because they're polygamous, were they also receiving other males?
No, so we made sure that so we raised all of them. So we had parental pairs, and then we raised them from juvenile stage on until they were sexually mature. So they didn't have any experience with any other males. So they're all virgin females.
But then after that, did they receive other males?
Oh, no. So we just let them hang out by themselves to lay eggs.
Gotcha.
Yeah, because then that complicates things, right? Then I'd have to like figure out paternity, which again is what I want to do in the future. But yeah, that's another excellent idea and makes total sense for the like literal next step. Yeah.
Sorry. That's going. Similar, related to that question,
Yeah.
do you know anything about if female choice or male, maybe not, female choice or male, courtship or anything is changed if the female is virgin or not? Like if she's mated with males before, is there
So yeah, in like other species, but in this species, I honestly don't think. The biggest indicator for male mate choice is female body size. Like, they are very interested in females that are large and that can produce lots of eggs. And then females too, they seem to not be super picky, even if they've already mated. And also, fun fact, the leaf-footed bugs will sometimes, the males will mate with females multiple times. So we'll see them go back, like, we had 3-hour behavior trials and we'd watch them and they would go back and mate again and again and again. So that was an interesting behavior too, that's like, what's going on here? Maybe it aligns with the post-copulatory competition. I don't know how long it takes for them to replenish sperm exactly, but 4 days seems to be like, if they've used their whole ejaculate, 4 days seems to be like, okay, they're fully recharged, ready to go. But maybe it's only a few hours. I don't know.